Hi everyone, I'm Ian Euler. I'm a graduate student at University of Idaho in Christine Parent's lab. And I would consider myself a microbial ecologist, but I'm going to try out some phylogenetic comparative methods today and see how much you all hate it. All right, so the topics I'm going to cover today are, first of all, the host microbiomes, or the host system that I study and where I study it, and why I want to add host-associated microbiomes to that system. And then I'll show you like a real brief sort of overview of the kinds of data that we find in microbial ecology, and then I will talk a little bit, or I'll talk the rest of the time about adding phylogenetic comparative methods to microbiome data. So the system that I work in is the Galapagos land snail system, genus Nasiotis. This is the most speciose system, animal system in Galapagos to date. They live all over the islands, and Dr. Christine Parent has actually been studying this system for over 20 years, trying to understand the sort of biogeography and colonization patterns of the islands. And so this is like a great system to add on microbial ecology because we know a lot about these snails. And so adding on these host-associated microbiomes, I'm interested in doing this for a variety of reasons. I'm sure if you've ever heard of microbiome talk, people are always like, oh yeah, they're super important for the host. They can do all these things. And there's a few other reasons too. Even in Galapagos, some people have started to see that there are some more antibiotic-resistant genes in the microbes in the guts of some of the reptiles in Galapagos, especially as you get closer to human-habited areas. We've also found in snails that some of these microbes could be useful for potential biofuels, cellulolytic microbes and things like that. And then there's some like cool questions about do our snails actually need a microbiome? Some animals, like the variation in how much a microbiome is important to a host. So these are the kinds of questions that I'm typically interested in. This study, basically, we got snails from these three main islands, which kind of represent the different island age classes in Galapagos. So on the left side of your screen, you can see this big island is the youngest island, or Fernandine is younger, but Isabella is the youngest island that I studied. In the middle there is Santa Cruz, and this is kind of the middle-aged island, and to the far right is San Cristobal. This is the older island. We collected over 200 snail fecal samples across these islands, and we also collected substrate samples to go along with that to try to like figure out the differences between the snails and the places that we found them. We did 16S rRNA and ITS using the Earth microbiome primers, and we sequenced those for bacteria and fungal sequences. This talk, I'm just going to focus on the bacterial sequences for now. All the data analysis was done in CHIME and R. And so don't worry about trying to figure out what's going on this page. Everything is very small. The whole goal of this was just to sort of show you the types of data that we get in microbial ecology. So you can see there's a bunch of PCOAs on there. In the bottom left, you can sort of see that sample type really separates well. So this is like substrates versus our snail samples. But as you look at things like between island interactions, it's a little bit more convoluted, and vegetation zone, it's almost totally overlapping. But one thing to note here is that we see these types of plots. We use things like Permanova to sort of distinguish differences between those groups. In the bottom middle, you can see a stacked bar chart because I'm a microbial ecologist, so there always has to be a stacked bar chart. This is just sort of to show you the types of the sort of amount of diversity that we're looking at. So this is at the order level. Each of these different colors is a different taxa. And so you can see that this data gets kind of complicated quickly. And this is, again, at the order level, it gets a lot worse as you go down to the genera. And then I just want to introduce the idea of differential abundances, because they'll come up a little bit in a second. This is just the idea that in some, like we'll split the data into pieces, and in some pieces, the microbes are more abundant in those types of samples compared to the other types of samples. So here you can see the blue bars represent microbes that are more abundant in the substrates, and the orange bars are things that are more abundant in your snails. My snails, I guess. Okay, so I wanted to add phylogenetic comparative methods to this based on the work of Sarah Hurd. I believe she is maybe the only person who sort of started this type of work. I don't know that anybody else has started it yet. I did see a paper that might do it the other day, but I haven't read it yet. But here we have, she did this in a group of tropical birds. And the main reason why we would want to do this is to sort of see, we know that these microbes are sort of developing alongside the host, or at least within the host. Maybe they have some kind of interesting evolutionary relationships with the host, and these types of methods could potentially be used to sort of uncover those sort of evolutionary relationships. And so I don't want her to fight alone. I want to do this too. Okay, so it's also a little bit interesting to talk about what is a microbiome trait if I'm going to be using these trait evolution models. And for this talk, I'm going to be talking about using just the relative abundance of certain taxa as this continuous trait that we can model on the tree. And so we're using this updated phylogeny from our lab as the tree, and then to choose which taxa to use, we made sure that they were at least in 75% of our snails. They are classified to family at least, and then they're differentially abundant from the substrate samples. And the reason we want this is just sort of these filtering is to try to get at the things that might be functionally important to these snails. We hope. Okay, so we have some predictions from the previous study that's done this. Basically, phylogenetic signal is pretty variable. You can kind of make predictions based on the spread of the data and how it looks on the tree. And so we get some microbes that have high phylogenetic signal and some that don't. So we sort of predict the same type of pattern. And then with model selection, they found that white noise was the most supported model, followed by Ornstein-Uhlenbeck. And so we kind of think, we predict that we also will see much less Brownian motion and early burst models when we do our model fitting. And so here's some preliminary results for this work. So phylogenetic signal, lambda. You can kind of see the names of the microbes don't matter that much. Well, I guess they do matter, but for our purposes, not as much. But so on the far right of the screen, you can see this microbe. This is actually a microbe that is not differentially abundant from the substrate samples. And so you can see that some of this phylogenetic signal could be driven by geographic differences in where the snails are. So that's just something to note. Also, we have a few things that have no phylogenetic signal, even though they're like ubiquitous across the snails, and probably do something useful. Things like Enterobacteriaceae is like a common gut microbe. I think one of the reasons why this doesn't have high phylogenetic signal is because it's pretty consistent across the tree, and so it's not got a lot of variation. One other thing to note before we move on is this, the middle bar, the highest one, is this chitinolytic bacteria, which could be helping the snails do some kind of like digesting of fungi or something when they're rasping the surfaces of their substrates. And so that's just kind of a cool result that this has got a lot of phylogenetic signal. And then the thing on the far left is Pseudomonidaceae. This is the most ubiquitous of all of the microbes that we saw in the guts. And so we're going to move on with this one because it's in every single snail, so we think it's probably a core microbe. Okay, so we did some ancestral state reconstruction. A couple things to note here. This is kind of a useful way to visualize the data. The top two over there, they're from two different islands. One is from the middle-aged island and one is from the oldest island. And most of these islands are sort of clumped together still, even on this ancestral state reconstruction. However, those two sort of like mixed a little bit. And they're also very high. And these are two snails that are actually arid zone arboreal snails, which is, so maybe there's something involved with their relationship with this microbe and having that sort of life history. Here's another way to view this ancestral state reconstruction. You can kind of see from this some of the patterns that are evolving here. You can see the very bottom group is the youngest island, and these have a very low amount of this microbe. These islands also had very high alpha diversity in general, maybe saying something about that their microbiome hasn't quite found its core microbiome yet. It's only a few 100,000-year-old snail. So we're not sure exactly what these patterns mean, but there's sort of some cool clues to move forward with. And then lastly, we have model fitting. And so here we have actually a very different result than the bird paper that has done this before. We do have the things that had a very low phylogenetic signal. Those are mostly explained by white noise, which makes sense. But the rest of them are more explained by Brownian motion, which is kind of cool, this sort of neutral evolution along the tree, a little bit more structure from the tree. And so this is kind of interesting to sort of see that we got some pretty different results from the other paper. So in conclusion, we got some pretty different results from this previous study. We still see a lot of variation in phylogenetic signal, but we do get a lot more Brownian motion model support. One thing to note is the bird paper used many orders of birds in their study, and this was one genera of snails, and so maybe that these snails are a little bit closer related, and so maybe their similarity along the tree has something to do with that. And then we got some cool new leads. So this Chitinophagaceae microbe, it has a lot of phylogenetic signal, and we think it could possibly be doing this chitinolytic activity for the snails. And then this Pseudomonidaceae is totally ubiquitous, found in every snail. It has a lot of phylogenetic signal. It's explained by Brownian motion best. And if you saw Andrew's talk yesterday, you might notice this as one of the things he clued in as a potential mucus cell symbiont, which would be really cool. I had no idea about that until yesterday. Okay, and future directions. There's a few other ways that we can measure these microbiome traits on the tree that I'm definitely going to use with this data. And those are things like just alpha diversity differences between samples. Also trying to get at functional information about these taxa. I think that my goal with this was to kind of treat this as a proxy for kind of testing these like functional groups. Like hopefully these things are doing a function for the snails. And so if we could actually get these functional groups, we could group them together and measure those on the tree instead. And so some ways that we could get those functional groups without with further sequencing is like shotgun metagenomics or metatranscriptomics, metabolomics for these kinds of functions. Okay, so hopefully people have suggestions because I would love to do this more. And thank you for listening. Sure. I mean, I think that there's a lot of, especially with this, there's a lot to parse out between like what in their environment is affecting the microbiome and what is actually tied to the host evolution. But absolutely, that could be something for sure.
Do you see any like alignment between the snail phylogeny and the phylogeny?
In terms of any individual bacteria, not yet. There's a little bit of like the thing people do in the literature, this phylosymbiosis thing where like microbiome similarity is like a similar shape as the tree. But yeah, not necessarily yet. But I haven't looked into it a lot, so maybe. Yeah.
So you had those groups of one microbe that was like in the environment, but also
Mm-hmm.
phylogenetic signal.
Yes.
Is there a way to extend this model to like include the environmental microbiome as like part of the method?
I think it definitely could. This first pass was just trying to be like, these are the microbes that are much less in the environment, but we should talk because I'm interested in like making it more holistic.